Today I'll be talking about tRNA, the role it plays with protein synthesis, and the adapter hypothesis. The adapter hypothesis was first proposed by scientist Francis Crick in 1956. Crick proposed the idea of a molecule that transports amino acids to the ribosome and interacts with codons in mRNA, placing amino acids in their proper order in protein synthesis. Transfer RNA is more commonly referred to as tRNA and is also known as the adapter molecule. tRNA can be thought of as a bridge between the genetic code in mRNA and the amino acids that make up a protein. Each tRNA attaches to a specific amino acid and carries it to the ribosome where the tRNA adds its amino acids to the growing polypeptide chain at the position indicated by the genetic instructions. Each tRNA has 3 nucleotides known as anticodon, which allows for it to bind to specific mRNA codons. The tRNA also has an amino acid attached to it, which is the same one that is encoded for by the codons to which the tRNA binds to. Here's a simplified structure of tRNA. You can see that there's an amino acid attached to tRNA, which is methionine, and has an anticodon sequence of UAC, which pairs with the mRNA that has the codon of AUG. You can see that the amino acid that is encoded by these codons is the same amino acid carried by the tRNA. But why do we call tRNA the adapter? The word adapter is defined as a device for connecting two parts as of different diameters of an apparatus. And we know that tRNA acts as a bridge between the genetic code found in mRNA and the amino acids that make up a protein. To be more specific, though, our bodies make proteins through a process known as translation. Translation is carried out in ribosomes, which are made of RNA and protein. Our bodies store specific instructions in pieces of mRNA, which are used by ribosomes to make proteins. tRNAs adapt the mRNA nucleotide sequence into the proper protein peptide sequence by acting as an intermediate between the two, which is why it got its name as the adapter molecule. The concept of tRNA being an adapter doesn't have to be complicated, though. We use adapters in our everyday life. As you can see, if we are trying to plug in a phone charger, we cannot just plug the USB into the outlet. We need an adapter to help us connect the two. Without an adapter, we wouldn't be able to charge our phone, and without tRNA, our ribosomes can't produce proteins. Mistranslation inhibits tRNA from acting as an adapter. tRNA is dependent on an enzyme called aminoacyl synthetase, which ensures that the correct amino acid is attached to the correct tRNA. When there is a mistranslation, tRNA will code for a different amino acid, which can lead to harmful consequences. For example, mammalian and bacterial cells can get heritable mutations. tRNA is very important for our everyday life. Without tRNA, our body cannot synthesize proteins properly, and without proper protein synthesis, cells cannot carry out their jobs.